Many people may be asking the question of why it has taken over 50 years for mRNA to reveal its therapeutic applications. Well, we are now immensely pleased and proud to welcome our next panel, which is going to take us into this fascinating question. And it is a great honor to welcome, to moderate this panel, the member of the Academy of Sciences and honorary professor of the Collège de France, Philippe Kourilsky. Philippe Kourilsky, the floor is yours.
Well, first I will say a few words about François Gros, to whom this round table is dedicated. And then, of course, we'll start the discussion. François Gros co-discovered messenger RNA in 1961 in the Pasteur Institute. There were two teams at the time. One was more molecular and was more genetic. François Jacob was on the genetic side, François Gros on the more biochemistry side, together with Jacques Monod. So the discovery happened twice, about the same time, with different partners, with different approaches, and have, of course, no time to discuss this. I think that François Gros, and I will mention that as one of his traits of character, was immensely kind and modest. And he told me many times, you know, have no merit in this because Jacques Monod had predicted everything. He had predicted messenger RNA in such details that we just had to find it, that's all. So that's actually one that he did. So François Gros had a, beautiful scientific career, but I would like to insist on his personal qualities because he was a man of immense value, and he was a man who was extremely kind. Many of us have benefited from his kindness and, of course, training, which has been my case. And he was immensely cultivated. He was really a man of culture in life sciences. He loved life sciences. He knew everything. Everything was interesting. So he was not only confined to some kind of organism and muscle, on which he worked a lot after working on bacteriophage and biochemistry. He was really open to everything. So he is a fantastic man. I'm really happy and honored to have this opportunity to pay a tribute, a small tribute, but a real tribute to him. So thank you, François, for everything. So now, ladies and gentlemen, we're going to start our session, and we will discuss a little bit the reasons why it's taken 60 years before this discovery came to something real in medicine and something enormously bright. But also, I think it's also interesting to devote much of a time to the future and to the, what is open, the avenues that are open, but also the difficulties that are before us and the reasons why we can be optimistic, but we have to be careful and understand the conditions for this development. Now, the panel is made of 4 eminent scientists, and will you introduce them, or?
Please go ahead.
Yeah.
You can do it, Father.
So I follow the alphabetical the order which is shown on this slide. You know, here it's a little bit like in Singapore. In Singapore, everything which is not allowed is forbidden. So, and vice versa. So I like to follow the order. So Marcel Hollenstein here is from the Pasteur Institute. And he will discuss mainly the overviews of the issues, advantages, examples of his project in Pasteur, namely in the field of chemistry of nucleic acids. Bruno Poddevin from DNA Script will describe the innovative platform applications. Regis Gervier from Sanofi, where he directs a large innovation platform, will discuss the examples of application. And Christoph Huber here is probably the most competent of us all because he is one of the co-founders of BioNTech and has a long experience of this issue and will provide us with some very important considerations. Now, the rule of the game is everyone has 6 minutes. Here, okay? Will control. I guess it's me, unfortunately. Or, okay. And so let's start by the order, which is here. And also let's start with you, Marcel. 6 minutes. Bang.
Oh, there we go. Better be quick. Well, good morning, ladies and gentlemen. First of all, I would like to thank the organizers, particularly Christian, for the invitation, and it's a real pleasure to be here to talking to you. Thank you, Philippe, for the nice introduction. So what I would like to do in these really 6 minutes is to talk a bit about the legacy of François Gros in the field of therapeutic oligonucleotides and also discuss the future developments in the field. Obviously the discovery of François Gros was fundamental because it allowed to refine the central dogma of molecular biology by providing a link between storage of genetic information in the form of DNA and protein synthesis. But beyond that, actually, François Gros gave us a fantastic therapeutic tool. Indeed, mRNA can be either a direct therapeutic agent in the form of mRNA vaccines, and this will be discussed by Christoph and Regis, but also in a more indirect manner. And in the field of nucleic acids, this can be done by two main approaches. First of all, you can use antisense oligonucleotides, which are short oligonucleotides that bind to specific stretches of mRNA, sterically block protein synthesis. Alternatively, you can use siRNAs, which are short RNA oligonucleotides that bind to pre-mRNA or mRNAs and silence these genes. Now, all these therapeutic agents have one common ingredient. That's why I'm a chemist. It's actually chemistry. They all contain chemical modifications. In mRNA vaccines, it's all the uridines are substituted with N-methyl pseudouridine to improve the translation efficiency, the yield of protein synthesis, and also to elude undesired immune response. In the field of other therapeutic agents, such as antisense and siRNA oligonucleotides, chemical modifications of the scaffold are absolutely fundamental to improve their base stability and their target affinity. Now, this is tremendous, and I think the field of therapeutic oligonucleotide has really improved over the last, for sure, 20 years with a lot of FDA-approved agents, but there are still some hurdles that need to be circumvented. Firstly, all these oligonucleotides need to be produced. So there can be two methods, essentially, chemical, by solid phase synthesis. So this is a very robust and reliable method for the synthesis of short oligonucleotides. You can upscale it to grams or kilograms. It's tolerant to modifications, but it's limited to rather short sequences. We're talking less than 100 or 120 nucleotides. Alternatively, and this is what is being used for mRNA vaccines, you can use enzymatic synthesis, mainly by in vitro transcription reactions using RNA polymerases. This we've seen with mRNA vaccines, can be upscaled, is tolerant to modifications. However, you do not have any control on the location of where you position your modification. Now, a number of companies, including DNAscript, and Bruno will probably talk about that later on, and academic research groups, including mine, try to push the development of new chemoenzymatic manners to push the synthesis of these chemical entities. A second important point is cellular delivery of oligonucleotides. So there have been major breakthroughs, especially thanks to the mRNA vaccines, in the form of delivery of oligonucleotides using these lipid nanoparticles. Now, this is fantastic. However, these lipid nanoparticles deliver the oligonucleotides to any type of cell, not to specific cells. There are chemical modifications, for instance, the GalNAcs, which allow therapeutic oligonucleotide to enter hepatic cells. But it has a very narrow scope. So what I'm trying to say here is that in terms of delivery, we have efficient tools. However, they're either too broad or too narrow of a scope. So here also, there will be a need for improving delivery of oligonucleotides. The last point is we need also tools, either bioinformatic or rapid tests, to monitor the in vitro and in vivo toxicity of therapeutic oligonucleotide, because this is still a handbrake in bringing oligonucleotides to clinical trials and eventually FDA approval. So we'll conclude by saying a few words, because I was here yesterday and there were fantastic sessions and a number of diseases have been mentioned. For instance, cancer, neurodegenerative diseases, antimicrobial resistance, orphan diseases. And I think by pushing forward these 3 points here, oligonucleotide therapeutic agents will also help in curing these diseases. Thank you for your attention. Hope I was in the 6 minutes.
Well, you did well.
Very good.
Good job. Mr. Poddevin, please.
Yes. So thank you, Marcel. I think Marcel gave some good insight about the complexity of how to make and how to use mRNA for therapeutical applications. And before, starting with my 6 minutes, I just wanted to thank the organizer to bring me here on the stage. But I have to say also that I was a bit surprised to be invited to join a discussion about mRNA, knowing that I spent my entire career making DNA. As a matter of fact, I'm now with DNAscript and not with RNAscript. And I was just thinking, okay, so they invited me. I am probably supposed to do something interesting about this conversation. And I thought, what kind of angle I can bring to this conversation. And I think the best angle is the only one I can give, which is the one I have from my professional experience in industry, in technology, and in DNA. And also to give, some kind of angle with regards to how technology is enabling and also can also be a bottleneck for the innovation and for, the advancement of therapeutical treatment. So the first thing, from a DNA point of view, the reality is that today, in order to make, or even to design or to discover mRNA vaccine, it all starts with DNA today. You need DNA in order to do the sequencing of the target. You need DNA to use it as a template so that you can produce mRNA. So DNA is everywhere. And the reality is that DNA has been everywhere for now 60 years. The first question we raised when we discussed about this roundtable is that why did it take so long to go from the discovery of mRNA and the understanding that mRNA could be used as a vaccine at some point, and 2020, when we really firstly use mRNA vaccine against COVID. And I think one of the answers, is not the only one, one of the answers is technology, one of the answers is DNA. In 1962, when mRNA was discovered, there was no way to produce synthetic DNA fragment. We didn't know about the chemistry, we didn't know about the technique in order to do that. In 1962, there were, I was not born, by the way, you know, I'm just saying what I was told. There was no PCR. There might be at that point, Philippe, you might confirm, maybe Maxam Gilbert sequencing with radio, no sequencing.
77.
All right, so I think the answer is clear. In 1962, we had no way to invent this wonderful technology. 30 years later, we had synthetic DNA, we had sequencing, not yet next-gen sequencing, we had PCR, but it would take another 10 years so that all this technology would become good enough, including next-generation sequencing, so that we could sequence the human genome. And when you take all this technology together, then you have the technical capability to turn the science and the knowledge into practical application. And according to me, that's really what happened, you know, between 2000 and 2020. It's, you know, the parallel progress on all these technologies that ended up to give you the practical technological capability to do mass sequencing, mass screening, design, and in order to end up to a functional mRNA vaccine, you have to do hundreds of cycles of design, test, confirm, design again, and if you don't have, you know, the technology to do that, it takes forever, it costs too much money. So technology is key. And if I just speak from my angle, synthetic DNA has been one of the key ingredients to make it possible. Now, what is the future about that? Synthetic DNA, DNA synthesis, was invented in the late 1980s. And the same technology has been used until now. No more progress. It was good enough to bring us where we are right now, but we might think that the fact that it was not further improved today might become a limitation. Why is it a limitation? When you want to manufacture synthetic DNA, you have very efficient chemical process, but they are chemical. So it uses a lot of hazardous chemical, you need to use high pressure, elevated temperature. That's clearly something you cannot do in your kitchen. So if you need tons of synthetic DNA in order to produce mRNA vaccine, you can only produce that in some kind of centralized factory. And as a matter of fact, today there is probably about 10 to 20 of such factories in the world providing synthetic DNA for the entire community, scientific and R&D.
Time.
30 more seconds. So what I believe is that we need to improve this technology as many others, and that's what DNScript is trying to do, to bring a new way to make synthetic DNA using enzyme and no more chemistry, so that you can really make this synthetic DNA in your kitchen, and in your kitchen means everywhere in the world. Meaning that when you will want to manufacture an mRNA vaccine in a very remote country, you will have at least this technology that will allow you to do that locally.
Thank you. Thank you very much. So we had research, then we had a startup, then we have industry, and then we have a mixture of everything. Okay. Mr.
Thank you very
Gervier.
much. And thank you very much, and thank you for the invitation today. I'm delighted to be in an environment actually where we can really co-chair discussion with some biotech startup here in this event, but also academic world. And it's all about mRNA actually. If you want really to push the envelope of such technology, you need to have a strong collaboration from the full ecosystem. So I have a challenge today because initially, effectively, when I saw the agenda, I was supposed to have a speech 6 minutes, right, with slide, and finally Philippe told us no slide, so I interpret no speech. But I will make it, don't worry. I will try to complement a little bit what Bruno was saying around the convergence of technology, acceleration of technology in order to reach really the ambition that COVID has been able to show us in order to really reach millions and millions of people worldwide. First element, you need to understand very well the biology. Philippe mentioned about it. 60 years to understand what mRNA is all about, how it can really bring benefit to the patient at the end of the day. But the biology itself, without technology, you cannot have a product. And that's why over the years and over the last 30 years, different technology has appeared, accelerated, in order to make sure that we can, down the road, really bring a product in a mass production, which is not a small piece of cake, to be honest, notably when you are working in vaccine with classical technology. When mRNA came, actually it's a dream for a guy like me who has made 27 years of CMC development, phase 1, phase 2, phase 3, and launch, tech transfer to the industrial world. It's a platform technology. So as soon as you talk about platform technology, you talk about acceleration of the development time, you talk about being far faster in patient, in human, and being able actually to bring new concept and new treatment to the patient. Now, the technology Bruno mentioned, it's one point, but at the end of the day, there is also what we have seen through COVID, right? Very, very efficient in a very quick manner to address unmet need, notably in a pandemic setup. But when you look a little bit what happened, notably in patient with mRNA COVID, the mRNA platform still need to be improved. And what we do in Sanofi, actually, thanks to the Center of Excellence that I'm leading and that we have built since 1 year, more than 400 people worldwide based in the US and France in order to address all the improvements that need to be put in place for such technology. It's very important to progress on this field. First of all, we need to understand what's going to trigger the long-lasting immunogenicity for such mRNA-based product. We know that with COVID, every single of us have received 1, 2, 3 shots, almost 4 for some people like me. And why? We need to understand what it's all about in immunology. We need to understand if it's specific to the target. Is it actually convertible to others, or are we going to see other type of behavior with new target? Are we going to be able to address bacterial disease? Right? Because today we know that it works very well for viral target, but not yet for bacterial. Another element that we are contemplating internally, it's also to make sure that we will be able, which is the beauty of such technology, to create combination of vaccine. How many, I would say, antigen we're going to be able to address at the same time? We know that there are several clinical trials on flu, quadrivalent, and potentially more than a quadrivalent one. But are we able to mix different types of disease? Is there any interferences as soon as you inject to patient? So all these type of things has to be cracked in order really to accelerate the launch of product. You have to pay attention to the time. One other element is really the stability. Stability, pharmaceutical stability, I mean, but also the mRNA stability once you are in vivo. There is many elements to crack here. Everybody is in a frozen form or lyo form potentially. I've not seen yet a fully liquid one. If we compare to very well-known vaccine like the pediatric vaccine that all our kids and even ourselves have received, like the hexavalent vaccine we are producing, Hexaxim, basically this product has 48 months of shelf life. 48 months. We are not at 6 months, right? So mRNA still need to progress on this dimension. Last but not least, reactogenicity. We know that all of us had here and there some reaction when we receive mRNA. We still have to understand how we can slow down a little bit the innate pathway linked to the composition of such product. Last but not least, as we go on the manufacturing side, I think, as you mentioned, Bruno, definitively we need to make sure that all the technology are mature in order to accelerate all the value chain, from the DNA synthesis to the mRNA production, the lipid synthesis, the formulation, the feeding, etc., etc. When you look actually for the mRNA production, I read a lot of papers saying that it's far cheaper to produce mRNA. Well, no. No, because the cost structure is totally different. Today, the enzymes are super expensive. The lipid remain expensive. The DNA, when you are doing it with classical technology, with some CMO, is very, very expensive. So we need to reduce the cost of all this raw material in order to make sure that down the road we're going to be able to be competitive or more competitive than we can be today versus classical techno. So I will stop here for the past a little bit. For the future, one sentence. You will never be able to master me. One sentence on the future. Definitively, mRNA platform will bring a lot of opportunity in many fields. In oncology, immunoinflammation, and you mentioned it already, and we will talk about it. And it's critical, again, to learn far better the biology around that, and also to develop new carrier in order to make sure that we can target specific tissues, specific cells. And I will stop here. Thanks a lot.
Thank you. Now, Christopher.
Well, first of all, I would like to thank Lady President, and being under the chair of Philippe Kourilsky is an honor and a pleasure indeed. After the thoughtful talks of my colleagues, you already know everything. What probably remains is I can put some color into the history of the development of mRNA drugs on one side, which has been French some 190 years ago. Mayence starting point was an immunotherapy hub. I found it in 2090. Basic research, discovery of tumor antigens, genetic modification of immune effector cells, stuff like that, tumor therapeutic antibodies at a preclinical level in a fairly sizable academic institution. We had, besides the collaborative research center, also a tumor vaccination center, probably the first in Europe, established 1994, something like that. We have used every antigen format available in these days, including, of course, peptides, proteins, multiple vectors, and we were not able to induce decent cellular immune responses against cancer cells, which would translate into clinical responses. We were quite desperate. And recruiting excellent new young scientists, we succeeded to recruit Hugo Shahin, who already was a famous scientist by the implementation of his Serix technology to unravel antibody-recognized, autologous antibody-recognized tumor antigens. Among other things he did, he's a technology leader, and he's a born leader. And Özlem Türeci, his wife, was extremely good in translating that into clinical studies. So they were the ones in 2000 under the roof of the Tumor Vaccination Center and the Collaborative Research Center, generously funded by the German Research Fund and the government, federal. They started mRNA drug development from the scratch on the shoulders of almost 2 generations of basic scientists. One has been very rightfully mentioned already. And the aim was, and that took something like 10 years in academia, to stabilize the information and to optimize translation and to make it more effective to enter the endoplasmic reticulum, to be processed and presented at the surface of antigen-presenting cells. And after something like 10 years, it was clear that one could amplify the potency as measured by induction of antigen-specific T cells by 4 to 5 orders of magnitude. Native mRNA didn't do the job, not in Mayence and not anywhere else. The optimized, the URA stuff did the job. And obviously there were other sites, University of Pennsylvania, with Drew Weissman, Kati Karik, who then joined our operation. So they were parallel developments. And when we saw the thing being mature enough, we founded BioNTech, which was not the first company we have founded. We had another unicorn doing antibodies, which are now entering the clinics. And in 2008, mRNA drug development was in the focus of the thing. And from then on, we did both fairly large-scale multiple tumor studies with different tumor antigens, among those neoantigens coded for by tumor mutations. And in order to do that, we had to digitalize and putting on a robotic platform that we can very quickly make vaccines on an individual basis, because mutational profiles are individual. So we had already the technology up and running for almost 10 years and vast clinical experience in the tumor vaccination field prior to the challenge of COVID. So we were ready to do that. And that is why we succeeded. And the other component was to partner with a pharmaceutical giant who is highly competent to do phase 3 studies. So that was the basis.
Bravo. This is very impressive. Thank you all. Now, what we'll do is to take questions from the audience, and I will, if you agree, keep my 6 minutes instead of 3 in the end, in order to try and summarize what's been said. The topic is enormous, and so we could spend several days on this because there is so much to say and discuss. So let's do that, and I will do my best in the end to capture what we might try to get out of this. So let's take questions from the audience. Who would like to ask a question to any of us? Well, any of the 4. Okay. So I will ask questions myself. Oh, there is a question. Ah.
Thanks.
Excellent.
I would like to understand better.
Please name yourself.
Yeah, Max Erzberg from Vidac Pharma. I like to understand better why is unmodified basis not effective in messenger RNA, and why you have to use pseudouridine in the sequence.
So that's for you, Christopher.
So, as you know, nucleotides induce strong cytokine release, in particular type 1 interferons. And interferon inhibits, for instance, antibody production. When you do prophylactic vaccines, you want strong neutralizing antibody production. We took the modification from Katikarico and Drew Weissman UPenn. We licensed that, and in a side-to-side comparison with Ugo's mRNA, we and others recognized that the vaccine response is better using the modified, the pseudouridine. The basic motivation to do the modification at the UPenn probably was different, because they were mostly focusing on protein replacement, substitution in hereditary disease. And there you can't have strong interferon toxicity responses. So it added up to a wonderful complementary development.
Thank you. Christian? Please.
If I may jump on that.
Marcel.
Just, there's one thing I just wanted to add, and there was a session on epigenetic modifications yesterday. You have to bear in mind that there are over 100 chemical modifications known in RNA in general. And actually, pseudouridine has long been considered as being the 5th nucleotide of RNA. So there's also a real interest in trying to modify RNA to make it more natural-like. But I think, like anyway,
Yeah.
Christoph had the good explanation. Thank you.
Thank you.
There's a question for
Introduce
BioNTech,
yourself.
Sorry?
Introduce yourself.
Oh, yeah. Christian Policard. I've been the executive committee of Sanofi a long time ago, and director at Institut Pasteur. And also, when I came as a CEO of Sanofi Pasteur, at the time the GV before the Pasteur vaccine were merged with the Emeritus vaccine, I did the merge, and the company was developing an hepatitis B vaccine, recombinant one, and I remember the people working day and night during a certain number of years, and to achieve at the end, success, But and when I saw what has been achieved, in less than 1 year, I have been very, very surprised. I am still considering what has been the reason why it has been so fast, because it had not be the case, it would have been really a disaster, worldwide. So, what are for you the factors, the two main reasons why it has been so fast? And the question for the future is, do you think that this experience you have had, at BioNTech and Sanofi has had also, do you think it will help in the future to develop faster biological drugs or vaccines or immunotherapies? Do you think that, because I've seen so many people changing their mind, partnering, for example, Sanofi and Glaxo. I leave it. It was a fight. We were taking people from the others and fighting. It has been a collaboration, and at BioNTech also you have been collaborating. So, what are the main reasons why it has been so fast? And secondly, what it will be used for the future to be better, to be able to address very fast new infectious diseases? So for me, I think it's very important.
Who wants to answer?
I know it's a very global question.
Yeah, I can start, and feel free to complement.
I have a few elements too.
So I think we have to realize that a pandemic is a real burning platform for everybody, right? And we know that when COVID hit beginning of 2020, all of us were really contemplating what would be the solution to address this situation. And we have to be cognizant on the fact that why it went so fast, at least from my perspective, is that all the government, the private company, the private institute put a huge amount of money on the table first to be able to accelerate in parallel all the tasks that are generally more sequential when you develop a classical vaccine. And it was billion and billion and billion of euro. So when you have to say that first, it would be difficult for every single program to put such amount of money, that's for sure. Now, we did learn a lot on what could we do differently in order to really accelerate the development of classical vaccine. Definitively, mRNA technology is one, because you have a real platform you can leverage as soon as you have the right sequence, the right vehicle, in order to accelerate to go to first in human. But there is also the progress made in many dimensions, in silico modeling, reducing the test in animals, but going more in vitro and having a really good test in order to screen very quickly the different solutions. And as you were mentioning, Bruno, it's also hundreds and hundreds of iterations in a very short period of time. So we do believe in Sanofi, and I do believe as the head of mRNA Center of Excellence, that we will accelerate the cycle time for new vaccine development. It will never be 1 year for every single target. But definitively, we should be able to reduce by 30 or 40% the classical time of development, definitively.
If I may add, one thing is, as you know, if you compare it with cellular expression factories, it takes overnight. If you do that for individual mutations on a large scale, as we did, you can really do a vaccine for a viral protein overnight. And that is what has been done on a regular basis in a GCP, GMP kind of network, where you do that every day. If you have ingenious leaders, and they are ingenious, really they are, it's good if the regulators help you. And they really did, in spite of all the critique you have heard, they were good. And everybody did his best. And then you had the giant partner, who is one of, as you know, the competent ones to do vaccines. You get wings. And if there is a request, and if the deaths of hundreds of millions are at stake, everybody does more. So it was a fortunate synergy in between all these things, but the technology was ready and it was digitalized and put on robotic. And that will change biotherapies. You can do any protein, and it's cheap, and you can modify, and it's target specific. It satisfies all the dreams, and it's taken up by dendritic cells on a large scale, almost selectively expressed in dendritocytes. So,
I'm glad that you mentioned the regulatory bodies, because they are often underestimated, very much criticized, but in that case, it seems to me that they have been great. The second point is that very practically, the clinical trials were easy, very easy. There were so many patients in developed countries, and so you cannot compare that to the clinical trials for HIV, for example, which for the vaccines have so far basically failed, where you had multicentric trials in 10 different countries and an enormous setup. That has been much easier. Okay, one last question before I synthesize. Yes?
Yes. Pierre Courteil, I'm a business developer in Abivax, French biotech. My question is that in the history of scientific and medical discovery breakthrough, we have many. We have vaccines, we have antibiotics. My first question is, where do you, at what level do you put the mRNA discovery and applications? And my second question is, do you think this was the last key we needed, the missing key to cure cancer?
I think it's easy to answer the second question. Okay, the answer is no. Okay, so if you don't mind. Now the first question, who wants to answer?
Well, I can probably try. Also, I have a biased view. It certainly will change the field. It certainly will become one of the lead technologies. And you can combine it, even what we do, for instance, mRNA vaccines to amplify the adaptive transfer of genetically modified immune cells. Again, orders of magnitude in vivo expansion, which you otherwise can do ex vivo only at huge expenses. No, it will become one of the businesses. I think it will be side to side with DNA. It's on the shoulders of DNA technologies, as we have heard. We start with a DNA template. One business will kind of help the other. And it already does, so we had a long-term exchange in that area. No, it will become something. And if we see that European scientists were able to advance that, we should think about European technology sovereignty, not in the sense of domination, but we should take advantage. We should talk to each other. How can we kind of synergize to advance at the speed other nations usually do?
Well, the time is unfortunately very short, so I will use the remainder 5 minutes to synthesize my understanding of what's been said. First, I would emphasize that 60 years is not unusual. It's taken 60 years for many, many developments. Remember just, for example, even the lasers resulted from very basic discoveries. And if we face now these 60 years, the proof of concept in animals started in the 1990s in several places, including in Paris, actually, because a fellow that you may know, Pierre Molien, had done some experiments, which was connected to transgene at the time, to sort of promote the idea. Okay. At the time, by the way, and several patents have been taken and forgotten, dropped, which also shows that it was not mature either in several other areas, including industry. But now I will emphasize maybe 6 factors, which I find important, and trying to recap what's been said. The first is that we need a lot of basic research, and not only in the principles, in the developments. For example, in the mRNA vaccines, it seems to me that the question of immune memory is just completely non-understood. And this is an area which is very important for many people. The questions of inflammation and cytokine production induced, blah, 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 all of that is very important, and all of that is basic research. So again, basic research. The second point I would like to mention is that there is an enormous amount of science to be done in the new therapeutic fields which are opened. So we heard about cancer, but this is not the only one. There are experiments about gene therapy which are just absolutely amazing. And if that confirms, it's a real second revolution in a completely different field. So mRNA is not restricted to vaccines. That's been mentioned by all the participants, but I re-emphasize it in my way. Now, several of the speakers have emphasized, and very rightly and very importantly, the importance of synthetic approaches. And so it's a tendency in biology that chemistry is getting more and more important. So we heard that from several of the participants, and this is extremely important for the future. Synthetic biology and biochemistry has a lot of future and is the future in certain areas. Now, I would like also to say that it seems to me very important to combine, of course, this is obvious, and we have a superb example in all the participants here, to come back academia and industry in terms of knowledge. There is a lot of knowledge in industry that researchers in the basic area do not know and superbly and stupidly ignore. So I like to use the terms, in French it goes better, lots of researchers do RANA. RANA is recherche applicable, recherche appliquée non applicable, which in English goes as applied, non-applicable applied research, okay? So a lot of people do that. They do experiments which have no future whatsoever because they do not know the constraints of development, which is totally stupid. So I would insist on this too. Now, I'm using my privilege of having discussed with my colleagues before to emphasize one point which is dear to several of them, namely the fact that it's very important to make the drugs available to everybody by special manufacturing systems that need to be automatized and sufficiently non-expensive to be spread in the developing countries as well as in the richer countries. Easy to say, but of course the economic forces and so on are there as well. But this is a very important for the future. And I know this is dear to the heart of Christopher, but all of you. And finally, I will also reinsist on the fact that there have been several books that have been written on mRNA, and reading these books makes it clear that a lot of innovation took place in Europe. Not all of it, of course, but a lot of the innovation took place in Europe. Now, what's been missing then is clearly the ability to invest. And the investment capabilities are absolutely essential at all steps. And we have no time to get through the various steps, but it's essential at all steps. And we need to really think of European systems to promote this. Again, not in an exclusive way, you know, we cooperate with others, the US, India, China, whatever. But the problem is that a lot of European innovation doesn't break through because the financial mechanisms are missing. And so this is another area which is illustrated by the story, the beautiful story of messenger RNA. And that is going to show more and more as the applications of messenger RNAs are going to spread in cancer and other areas. It's urgent. So we really need to make some kind of European effort urgently. So, sorry, we left the field of science to go to finance, but finance is important. By the way, I recall to some of you that the financing mechanism have been invented in several countries, but French played a very important role in the United States to invent capital risk. Actually, it was invented by the French, but in the United States and not in Europe. All right, so I think, again, we were short of time to discuss an immense field, but at least I hope we open for you some avenues of thinking and even some understanding of some of the problems. So I would like to thank you all very much for your participation and thank the audience and the remote audience. And we finish in time.
Thank you for your majestic, magnificent moderation and a remarkable roundtable. Thank you.
So, as the chairman of this event, I would like very thank you for coming at this event at HCD for this fourth event. Thank you very much. It was a great roundtable, and it was important to have a thing to Mr. Gros, and so we were very pleased.